Hello everyone, I'm Tanuja Kopal, Consulting Editor for BioCompare, and welcome to Bench Tips webinar. Today we'll be discussing some of the new developments in next generation sequencing and how to better optimize some of the sequencing workflows. Bench Tip webinars were created to bring together young scientists, postdocs, senior graduate students who spend a lot of time at the bench fine-tuning and troubleshooting different assays and techniques. We also bring experts in the field as moderators, and they are the people who spend a lot of time developing and designing some of these assays and experiments. So some of this technical knowledge remains buried in lab notebooks or buried in the materials and methods section of research papers, and we thought that the Bench Tip webinars would be a good forum to bring together all those ideas, share some of those learnings, discuss what worked, what didn't. So that's why we are here for you. But before I begin, I want to extend a big thank you to many of our sponsors who are making this event possible, and for the team at BioCompare for putting this together. So our sponsors for the webinar today are 10x Genomics, Beckman Coulter, Selecta, Eppendorf, Levitas Bio, and Roche. Thank you so much for making this possible. And before we get into the webinar, I want to remind everybody that this webinar is meant to be informal, informative. We want to hear from you. So after our short 3 talks, we will have a Q&A, a question and answer session. So send in your questions and comments by clicking on Ask a Question button, which is on the upper right-hand side of your screen. And remember, don't wait till the end to send those questions and comments, because very often we run out of time and some of those thoughtful questions that you send us don't get answered. If you have any question, technical question, just click on Test Your Connection button, which is at the bottom of your screen. From there, you can get any support that you're looking for. You will also notice that on the right-hand side of your screen, you have an overview of today's webinar, and you have information about our speakers. And you will also find at the bottom social media widgets for you to share this webinar with your friends and colleagues who may find this beneficial. So with that, let me welcome our moderator today, who is Dr. Mandovi Chatterjee. Mandovi did her PhD in cell biology at University of Virginia. After a brief postdoc at the Brigham and Women's Hospital, she joined the single-cell core at Harvard Medical School, first as a postdoc, and then she was a deputy director of the core. And very recently, she has been promoted to director of the single-cell core. So congratulations, Mandovi, and thank you so much for joining us today to share some of your learnings.
Thanks, Tanuja. And hello and welcome to today's webinar, which is going to focus on the practical aspect of doing single-cell experiment. In the last 10 years, single-cell experiment has proven to be useful in determining a highly resolved cellular composition in complex tissue systems. After this webinar, hopefully you will have a better understanding about the workflow of performing a successful single-cell experiment with knowledge about available technologies, protocol choice, and quality control. This is a very popular analogy to understand what single-cell biology means. Bulk RNA sequencing, which has been in existence for quite some time now, looks at the average gene expression across a particular tissue system. Bulk sample analysis is like putting a fruit salad in a blender, and the taste is an average of all the ingredients. Whereas analyzing single cells is like tasting individual fruit to gain a much more detailed understanding of the composition of the fruit salad. Bulk RNA sequencing is useful for comparative transcriptomics or determining disease biomarkers, but it is unable to capture the heterogeneity of a tissue. In a complex tissue system, the constituent cell types and their relative abundances can be determined only using single-cell RNA sequencing. One of the most popular questions that people have asked using single-cell RNA sequencing is how heterogeneous their tissue of interest is, and the quest is to discover a new rare cell population. Now here I'm talking about the heterogeneity within a tissue. Now single-cell RNA sequencing has enabled us to look at the variability in the gene expression within a particular cell population as well. Moreover, With the advances of single-cell RNA sequencing, it is now possible to build a detailed cellular atlas of complex and dynamic biological processes, including developmental trajectory and time course experiments. In the last 10 years, the field has progressed significantly, and there are different single-cell platforms and technologies available now. Here are a few of them. The well-based methods such as SmartSeq are low throughput and labor-intensive, but full-length transcript can be studied using these platforms. On the contrary, the nano or microwell-based platforms or the droplet-based platforms offer high throughput and are cost-effective too. Now, how would you choose which platform to use? That really depends on your experiment. If you're looking to detect a rare population of cells where you would need to profile a lot of cells, you will have to go for high throughput platforms. But if you're limited by the starting material or if you're interested in looking at full-length transcripts, low throughput methods would be the way to go. Now, low throughput methods offer high sensitivity too. By high sensitivity, I mean the number of RNA molecules that you can detect. And throughput refers to the number of cells that you can profile using a certain platform. More RNA molecules can be detected using low throughput platforms, whereas higher the number of cells, lower the sensitivity you get. However, there's been a surge of using high throughput platforms recently, since their availability, partially because of their cost effectiveness. Now, what I have here is a very simplistic view. The reality is actually more complicated than that. But regardless of the platform you choose, the basic steps are the same. First, you need to generate a good quality single-cell suspension, followed by cell barcoding, making sequencing ready libraries, then sequencing them mainly by next-gen sequencing platforms, and then analyzing the data. Now, sample prep is unequivocally a critical step in order to get good quality data. For single-cell RNA sequencing, high-quality sample is a must. What do I mean by high-quality samples? The samples need to be highly viable. More than 90% viability is ideal. The cells need to be alive up until the point they're barcoded. The cells should not clump, and little or no debris is recommended. Now, does this mean that we do not run samples that has lower than 90% viability? No, that's not true. We do run them. But what happens when the viability of your sample is low? So when the cells die, they lyse and release their RNA content in the sample, which is represented by this light green color here. And these free-floating RNAs get barcoded as well, leading to high background in the data. So more free-floating RNA in your sample, more background you get as depicted by this plot on the right. Now, if these free-floating RNAs dominate your library prep, you end up wasting a lot of sequencing reads for something that eventually will be eliminated during analysis. So in order to obtain a high-quality sample for single-cell RNA sequencing, it is important to optimize a dissociation protocol that is well-suited for your tissue of interest. You're often in the best position to figure this out because probably nobody knows your cells better than you do. I prefer running a pilot experiment before jumping into a gigantic 32-sample 1 million cell experiment. A pilot run gives you a lot of valuable information, starting from how good your sample prep is to what sequencing depth is optimum for your tissue of interest. Now, short sample prep time and maintaining low temperature during the sample prep has been proven to be beneficial. Gentle lysis condition and centrifugation, short fax time, sometimes with using bigger nozzle help. Removing debris from the sample is crucial. And having BSA or low percentage of FBS in the final buffer keep the cells happy as well. So one of the questions I often get is how many cells should I analyze and how deep should I sequence? Unfortunately, there is no straightforward answer to that. It really depends on the question you're asking. For rare cell type, of your interest, you need to analyze many cells. And for overall heterogeneity, fewer cells might just be enough. As a rule of thumb, you need about 50 to 100 cells with unique transcriptome signature to form a distinct cluster. That means if you profile 5,000 cells, about 50 or 100 cells make up to 1 to 2% of the entire population. If you want to see smaller subpopulation than that, say 0.5%, you will need to profile double as many cells, and so on. Now for sequencing depth, rare the transcript, higher should be the sequencing depth. And deeper the sequencing, higher is the cost. So there is a fine balance that you have to strike. So sometimes people start with shallow sequencing depth and then decide how much deeper they need to go after looking at the data. Now, batch effect has been identified to be one of the challenges in single cell data. The batch effect can come from many sources, starting from sample prep, animals from different clutches or litters, different batches of reagents, even operator, library prep performed at different times, etc. There are certain ways to minimize the batch effect as well, if not completely eliminate them. There are wet bench solutions, such as using biological replicates, a same batch of reagents if possible, keeping the operator same, prepping libraries together, sequencing them together, and so on. And recently, pooling samples by sample barcoding has become pretty popular as well. There are a few dry lab solutions as well for batch correction. Sometimes batch effects are unavoidable. For example, in case of patient samples, time course experiments, perturbation experiments, and so on. With this overview, it's time to move on to the next part of the webinar. And it's my pleasure to introduce our two speakers today, Dr. Thomas Kraus-Gruber and Jonathan Katzeneln-Bogen. Dr. Kraus-Gruber is a postdoc in Dr. Christoph Bach's lab at Research Center for Molecular Medicine of the Austrian Academy of Sciences. He's a system immunologist working on structural cells and studies their contribution to immunity. His research uses high-throughput omics technology and functional assays to study complexity of organ-specific immune functions. And our second speaker today is Jonathan Katzeneln-Bogen. He's a senior graduate student in Dr. Ido Amit's lab in the Department of Immunology at the Weizmann Institute of Science, Israel. He has specialized in developing single-cell RNA sequencing technologies for immunogenomic research. He is interested in deciphering the molecular landscape of tumor-resident myeloid suppressor cells for the discovery of new targets for immunotherapy. From their talk, we will hear about the single-cell technologies that they applied for answering their questions about their findings and the challenges along the way.
Thanks for the introduction and thanks for having me at this webinar. Today, I would like to talk about one of my projects, which is called structural immunity, where I look at immune functions beyond hematopoietic cells. And within the next 10 minutes, I really would like to showcase the power of multi-omics profiling on sorted bulk populations. But before going into my project, I really would like to discuss the distinct modalities that can be captured by sequencing. And actually these days, almost anything can have sequencing as a readout. And hence, I would like just to go over 4 modalities that are often used. First of all, and the new kid on the block, is sequencing of surface receptors. And this enables us to go beyond the colors that are available on the flow cytometer and really look on the quantitative and qualitative way of the expression of surface markers on cells. Secondly, we can look at the transcriptome and, for example, analyze the genes that are expressed at steady state in a cell and compare this once the cell has been activated or perturbed. We can also look at the occupancy of transcriptional regulators to really find out which proteins and transcription factors are important for gene regulation. Furthermore, we can look at the epigenome and, for example, look at methylation profiles or open chromatin regions to add yet another complexity to gene regulation. And obviously, all of those modalities, and also the ones I haven't listed, can be looked at in bulk resolution and single cell resolution. And as you just heard from the introduction, both of these methods have their pros and cons. However, I would like to show you that it doesn't have to be always single cell resolution. One can also learn novel biology by looking at bulk samples. Now, as an introduction to my project, if we look into a textbook and we want to know about how immune responses are regulated, we will usually learn about two major events. First is the activation of innate immune cells, which is followed by the activation of adaptive immune cells. If mentioned at all, structural cells such as endothelium, epithelium, fibroblasts would be considered merely a physical barrier. However, research over the last couple of years have clearly indicated that some of these cell types can have profound organ-specific immune functions. Take, for example, epithelial cells in the gut and in the lung. However, no one looked at those three cell types within and certainly not across organs in terms of immune function. And this is what we wanted to do. And the way we did this is we used a systems approach to analyze structural immunity. And here we had to first optimize the isolation of 12 organs of the mouse to get out structured cells. And this was very important because organ dissociation methods are usually geared towards getting out immune cells. However, what we found is that those methods would have a negative impact on structural cells viability. Hence, I optimized the protocol, a single protocol that I could use for all those 12 organs, resulting in high quality and good viability structural cells. And actually, this is very important if you have sequencing as a readout, that you really want to have good, high quality material going into your sequencing, because this greatly enhances the possibility that you will get out good and high quality data out. Out of those single cell suspensions of the organs, I used sort purification to sort out endothelial cells, epithelial cells, and fibroblasts. And because I was analyzing my cells in the bulk population, it was still very important to have a good sort purity check. So this can be either done by flow cytometry or, for example, as we did, bioinformatically. Both are reasonably valid. On my sorted bulk populations, I would then perform flow cytometry, transcriptome analysis. I perform chipmentation, which is a form of chromatin immunoprecipitation, and then I was in particular interested in the histone mark H3K4 dimethylation, which is a mark for active promoters. And I looked at open chromatin regions using the assay ATAC-seq. I profiled structural cells at steady state and perturbation and kind of wanted to answer three main questions. First of all, I wanted to know the role of structural cells at homeostasis and during inflammation. Furthermore, I was interested in how structural cells would communicate with hematopoietic immune cells. And in order to answer those first two questions, I primarily used data generated by flow cytometry and transcriptome analysis. However, I was also interested how immune regulation in structural cells would differ across organs. And here I used the data from a chipmentation ATAC-seq experiments to really find out which transcription factors ultimately implement an organ-specific and cell type-specific immune functions in structural cells. Today I will not have time to go over the results of all of our data. However, if you're interested, here's a link to our paper that we published last year. Now, going into the data, looking at our transcriptomic data, we found that structural cells express key immune regulators, and those would mediate communication with the hematopoietic system. So what we did here is essentially we took our transcriptomic data from the structural cells, and we used public available data on immune cells to draw up an organ-specific and cell type-specific cell interaction network. And what we found is that structural cells would interact with the immune system in an organ-specific and cell type-specific manner. And we actually think that this is one of the major ways how structural cells can influence an immune response, by interacting with the immune system. However, the true novel biology that we found is when we combined our transcriptomic and our epigenomic data. Because when we integrated our data, we found genes with an epigenetic potential, and those showed up as outliers in the correlation between the transcriptomic and epigenomic signals. And what I mean by that is depicted here in this graph. So usually we would expect, if there's high promoter chromatin accessibility, that there's also high gene expression. In this case, we would talk about the realized potential of a certain gene. This is in contrast to low promoter chromatin accessibility, where we expect that there's also low gene expression. What we found in structural cells is many genes, and among those many genes with immune functions, that showed a high promoter chromatin accessibility, but relatively low gene expression. And those genes we term to be of unrealized potential, because in theory they would be ready to go, but are just not transcribed yet. And as an example, I would like to show you liver endothelial cells and liver epithelial cells. And in those two cell types, the gene interferon gamma receptor 2, depicted here in red. In liver endothelial cells and liver epithelial cells, we detect in both cell types high promoter chromatin accessibility. However, in liver endothelial cells, this is coupled with relatively low gene expression. So in this cell type and in this organ, we will talk about an unrealized potential for interferon gamma receptor 2. This is in contrast to liver epithelial cells, where high promoter chromatin accessibility correlates really well with high gene expression. So in this cell type, we would talk about a realized potential gene. Now, seeing this unrealized potential, we hypothesized that actually different aspects of this potential can be triggered by different stimuli. And in order to prove this, we focused on viral infection. And here we used the acute viral infection model. On this model, the peak of disease is on day 8. And on this day, again, we harvested 12 organs of the mouse, and of each organ, 3 structural cell types, and we performed transcriptome analysis. And then we asked the question, how many of the genes that we determined to be of unrealized potential of steady state would be activated upon the acute viral infection? And what we found is that there's a cell type specific and organ specific activation of this immune potential. And especially in liver and lung fibroblasts, but also in spleen endothelial cells, almost up to 50% of the epigenetic potential is activated upon the acute viral infection. Now, as I told you, I don't have time to go over it, but essentially the key results of our study is that structural cells are organ-specific mediators of host immunity. We found there's an organ-specific immune adaptation of endothelium, epithelium, and fibroblasts, that there are complex interactions between structural cells and immune cells. And most importantly, we identified an immunological potential of structural cells that is encoded in the epigenome. And in our mind, there's 3 entities to our immune system. There's structural immunity, innate immunity, and adaptive immunity. And I hope with my presentation, I could show you that multi-omics profiling of bulk sorted cell populations is a powerful tool to discover novel biological insights. And with that, I would like to thank all the people in Christoph Bock's lab at SEM in Vienna, our collaborators, and our funding bodies. I would like to thank you for listening and look forward to answering your questions.
Hello, everyone. Thank you for the nice introduction. I want to talk to you about today about single-cell genomics and how it became a stepping stone for future immunology research in recent years, and to present a recent development in that field. So before we start, I just want to just a quick overview about the differences between cell types and cell states. Just a quick overview for us to be on the same page. So imagine that this box you see here is the full playground of the cellular phase. So in this playground, you can have, this is the most omnipotent cell within your body, and there are all the fates are inside here. So during development, we know that this potential is being narrowed down, right, throughout a series of commitment until we're reaching a point where we have more or less stable cells. In our case here, this red cell and this orange cell, which are quite stable, meaning that the red cell will never be orange and the orange cell will never be red. And of course, we have a lot of lineages and we have a lot of different cells, and this fixed and stable cells are cell types. And these cell types are quite interesting, and we can use them answering a lot of very interesting biological questions. So if this is the cellular framework, we have again a lot of cell types. But we know that answering biological questions, interesting ones, using only cell types doesn't give us the full picture, because let's look at, again, this red cell. This red is not just plain red all the time, right? We have a lot of flavors of red in different tissues. So every cell type has different flavors. So we can have, for example, one cell state, which is let's say, this dark red in a specific tissue, and we can have like a light red, which is another cell state, again red, in another tissue, and it's true also for the purple one. Okay, so we have a lot of flavors within these cell states, flavors which are representing cell states, which are more flexible, induced by the surroundings, and sometimes even reversible. And these states sometimes are very, very interesting, again, to answer interesting biological questions. So when we do bulk RNA-seq, as Mandovi said at the beginning, and I use this example again, let's say that this red cell is, you sorted, you stained it extracellularly. These are, let's say, CD8 T cells. So you sorted CD8 T cells. These CD8 T cells, these red ones, have a lot of different flavors within the tissue, but you sorted all of them. So when you do bulk RNA-seq, what you do is averaging again the mRNA expression, and you're getting this plain, this flat red at the end. Okay, so you cannot detect which mRNA molecule came from which cell state. The same is true for, let's say, these are CD14 monocytes. It has different states, but when you bulk show it again, you lose this complexity at the end when you analyze your data. Using single RNA sequencing, we're doing the quite opposite. This is a bulk resolution. In the bulk resolution, we see this square plain pink. When we zoom in, we actually see that this pink is not just pink, right? It's composed of different colors. It's much more complex, and this is exactly the resolution we're focused on in our lab. Okay, so this is exactly what we're doing in our lab. We having this protocol of plate-based single-cell RNA sequencing, we usually get single-cell suspension. We stain the cell, then we sort them, produce these libraries, as Mandovi mentioned, and analyze the data afterwards. And what we do when we want to define cell identity is actually we want to accumulate many layers of information regarding our desired cells, right? And we can describe these same cell using different layers of information, depends on what we're gathering, right? What we're collecting. So we can define cells regarding according to their, for example, transcriptome. This is what we do. We can describe this same cell according to its lineage or morphology or location within a tissue. So we can define the same cell or the same tissue using different methods. And the interesting part is to combine them to get a more holistic view about the cellular identity. So this is what we're trying to do in our world, combining transcriptome and proteome. This is quite intuitive, and we do it regularly. What do I mean? Usually we stain the cell. We can stain surface markers. We stain the cell, and then we can, when we're using the FACS, regularly we can sort them according to a certain expression of surface protein. The problem is that surface markers are only 30% of the total proteome. So we're actually, in our case, blind to the rest of the 70% of the intracellular world, which is quite interesting. What do we have there in this intracellular world? So we have signaling events that we're totally blind to. I mean, we cannot detect them. If you want to do RNA-seq and monitor some signaling event. We have transcription factor expression, which we also cannot reach. And we have a lot of metabolic elements that are in there that can be quite interesting. So this world is very interesting. And the reason why it wasn't accessible for us was that there was a trade-off between intracellular staining and single-cell RNA sequencing or RNA sequencing. I'm sure a lot of you who try to do it knows it's quite challenging to fix and permeabilize the cell, which is necessary for intracellular staining, and then having good RNA quality. So you had to choose what you want to do. You want to do this or this, you couldn't do them both. That's why we came up with INSET technology, which stands for intracellular and staining sequencing technology, which is quite straightforward, quite simple. We developed a series of buffers. You can do like a regular intracellular staining, exchange some buffers, and move forward with these cells for single-cell RNA sequencing, and then combining these two layers together, and it provide a very interesting insight in many fields. That's for you to appreciate the performances of this methodology, we have here fresh human blood cells, PBMCs, and insect-treated, meaning fixed PBMCs, and you can appreciate that we have similar, same composition in both cases, so we're not losing any information in that sense. I'm not going to go over everything. We had a lot of different results there. You can find it online. I just want to say that we demonstrated the ability of our methodology to target some signaling events, defining responders and non-responder cells to FDS stimulation by targeting phospho-P38, and it was quite interesting. We also targeted some transcription factors from T cell population within human blood, so we produced this transcription factor map, isolating cells to the expression of a specific transcription factor, then profile them using single-cell RNA sequencing. It was quite interesting. We had beautiful maps there, 2D maps that you can appreciate here. But what we were mainly interested was detecting and try to profile the myeloid suppressor cell population within the tumor microenvironment. So this population, this subpopulation is quite elusive, and it looks very much like from the outside like other cell types, like other macrophages, monocytes, and it's very hard to isolate it. It's quite hard because it doesn't have a distinct surface marker. But what they do have is a very distinct metabolic signature, mainly characterized by increased arginase 1 and other factors, but increased arginase 1. So we said, okay, we have the ability to do it. Let's stain for arginase 1 and sort arginase 1 positive and negative cells, and this is exactly what we did. And I want to highlight two findings that we had there. One was we came up with this result of the high correlation between R1 expression and TREM2 expression. TREM2 is a membrane protein, which is very interesting for us because we have encountered this protein before in Alzheimer's disease, so it was very interesting for us. And the other thing that we defined was a unique subpopulation of, we call them MREG, which stands for regulatory macrophages, which were there and were highly RG1 positive cells, which can be characterized by increased expression of GPNMB, which is another surface marker. Then we said, okay, is this just a correlation to TREM2, or TREM2 indeed have a specific function? So we took it to test the functionality, and we produced a TREM2 knockout mice, and then we injected tumors. And as you can appreciate here, TREM2 knockout mice demonstrated reduction in tumor size, suggesting that there is a role for these TREM2R1 cells. And when we molecularly identified what's the difference between wild type and TREM2 knockout mice, we indeed saw reduction in these regulatory macrophages, MREG population. We believe that it led to reduction in CD8 dysfunctional T cells and elevation of the cytotoxic T cell NK compartment, which led eventually to the reduction in the tumor size. This is my last slide. I just want to emphasize and conclude the insect strengths as I see them. First is targeting signaling events, because again, we're totally blind to signaling events when the readout is only RNA, only transcripts. So it's a very powerful tool for that. The second thing is that we all know that there is not a 100% correlation between the expression of RNA and protein, so we can evaluate this correlation using intracellular elements as well. And the last and very important issue here is the enrichment of rare populations. Sometimes the population you're interested in are very rare, and you want to enrich for them, and the only way to do it is by targeting some intracellular element which is exclusive for them. And it's true mainly with the clinical samples where the total amount of cells are usually limited. So it's a good way to do it as well. I want to thank Fadi, Adam, Asaf, and Ido who led this project with me. This is the other lab members who helped us. I want to thank you for listening, and I'll be happy to take questions as well.
Thank you all so much. Mandovi, you did a fantastic job just setting the stage for us. And Thomas and Jonathan, appreciate you sharing your recent findings and also walking us through the process of why you did what you did. So with that, I think we should open up the floor to questions. Again, a quick reminder to our audience that you can send your questions and comments. Just click on Ask the Question box, which is on the right-hand side of the screen, and type away. And we will get to your questions right now. We have a lot of time to get to them, so I'm hopeful that we will be able to address most of them.
Thanks, Thomas and Jonathan, for a great presentation. So we have a few questions already. So one of the questions is, do you have recommendations on RNA-seq analysis platforms? Jonathan?
We're using an in-house analytical platform, so I'm not that much familiar with other things. We use it's called MetaCells, and the main principle is that it clusters together cells that looks the same, and we're actually working with the single cells. At the end of the in the analytical part, we're using things that we call metacells, which are a bunch of cells, usually some like dozens of cells that are express more or less the same genes, and we use them as we treat them as one cell. But we're using an in-house technology. I'm not that familiar with other technologies.
Thomas, do you have any comment here?
Yeah, similar to Jonathan, we also use like in-house technology, but essentially what we mostly look at, for example, in bulk RNA-seq is just differential gene expression analysis, and I think there are plenty of either softwares that can do that, or you can also find online, if you look at Git repositories, I'm sure you can find the relevant code to do this type of analysis.
Mm-hmm. It's a great question. We didn't get a chance to cover data analysis today, but it is a big component of any single cell or bulk RNA sequencing experiment, and one needs to set aside adequate resources for that in terms of time and money. So if you're well versed in bioinformatics yourself, there are some open access resources out there, like Thomas mentioned, or you may want to have a bioinformatician to do that for you. Moving on to the next question. How do you decide which technique to go with, single versus bulk sequencing? And is there a decision tree that can guide you, Thomas?
Well, I think one major thing to consider is obviously money, because obviously single cell technology is still very expensive, and I think you need to think careful if this is the right assay for you. But essentially, if you expect there to be a high cellular heterogeneity, then obviously a single cell approach can help you to untangle this. However, if you are already interested in a very particular cell subset, and you are able to define this subset very well with, let's say, surface markers, then obviously you can sort purify those cells and analyze them in bulk. So I think based on decision tree, I think it would be very useful to speak with someone who has experience both with bulk and single cell technology, and then help you guide through the process.
Yeah, I completely agree with Thomas on this, that there is there's a sort of an attraction towards adding a single cell experiment in your paper, but what people might want to keep in mind is that the sensitivity of bulk RNA sequencing is a lot higher than single cell RNA sequencing, and the data to noise, signal to noise ratio for single cell RNA sequencing is much lower than bulk RNA-seq. So there is certainly a lot of value in bulk RNA sequencing as well. given, like in certain conditions.
Right. And Mandori, I was curious, being, with a core lab, like, do you typically sit down with your users even before they start the experiment and kind of figure out if they have actually looked at all the different possibilities?
Yes, absolutely. I mean, we at our core facility, we value consultation a lot before actually getting into the actual experiment. We start with discussing, we covered a lot of general topics in this webinar today, and but then it's kind of a very personalized experience for every single person. What works for you may not work for somebody else, so it is important to look at your particular experiment, and so that's where we start. And depending on the particular experiment, we suggest what platform would be best, how many samples would be necessary to sequence. And given the single cell experiments are very expensive, often the decisions are guided by the funding situation of the lab as well. And we consider those too.
I see. No, that's useful to know. Yeah, thank you.
So our next question is, can you elaborate on the sample prep issues, especially for rare populations? Jonathan, do you want to comment on that?
Yeah, I want to comment on the tissue prep maybe, and maybe I will focus on the rare population issue because I think it's one of the strengths in the technique we use in our lab, which is plate-based. So I think the most intuitive thing to say in this matter is that sample prep is one of the maybe the most important part of the entire experimental design. First, because it's because we're dealing with like highly delicate systems sometimes, and we're trying to catch even one or two molecules. For a very small and tiny thing like a cell, it has to be very precise, and each tissue behaves differently, and it needs to you need to have to be very careful with which protocol of dissociation or preparation you're doing in each project and in each tissue. Even I'll give you an example from the last few months. We're struggling with, for example, now we're struggling with preparing blood samples in our lab, a tissue that seems to be like highly, very easy, just like a very everyday thing like people do in their labs. We're struggling with this because it appears that they tend to be very delicate in some conditions, and we're having a hard time extracting valuable data out of them. So you need to be very precise with how you're preparing your sample. This is one thing. And regarding the rare populations, some dissociation protocols tend to enrich or deplete different subpopulations. It's very hard to benchmark it. But it's something you need to consider. When we start a new project with a new tissue, we usually collect many dissociation protocols and we compare between them, and we try to see which dissociation protocol gives us the highest heterogeneity of the tissue. When you want to target rare populations, it's highly important, and regarding like the platform we use because we can do it with the plate-based technologies. We can pre-select them. If it's very rare and you're just run it with the microfluidics technique, you sometimes won't have like good representation of your desired cell type. So when you use plate-based technologies, it's quite optimal to use them when you want to target some area population, because then you can isolate them from the tissue with flow cytometry.
So Jonathan, you sort them yourselves, right? So how stringent are your sorting conditions? Like how many markers you use? How many markers do you use to select for these rare populations?
So it depends. We recently purchased a new fax machine that we can incorporate many fluorophores, but whoever works with fluorophores know that it's like you have compensation problem. It's like quite limiting in that sense. You cannot like integrate many markers, like not dozens. You can integrate, I don't know, we use like 10, maybe 12 markers at a time. And when we want to hunt down some specific cells, it might be quite tricky. Especially when we're dealing with the clinical samples. So when you do, if you're working with mice, you can accumulate few mice together or whatever, and then you can like have a sufficient number of cells to work with. But when you have like rare clinical sample and you want to hunt down rare cells, it can be quite challenging, and almost I think impossible to do it with microfluidic systems.
Correct, I agree. But then while using a very stringent condition during fax sorting, do you not, or have you run into biasing your sample?
What do you mean?
Like, for example, I mean, I personally prefer using a more broader marker compared to using a very stringent sorting population, because my worry is that while choosing like multiple markers for one single rare population, you might lose a lot of cells which might be relevant.
Yeah, I agree. You better be like broader than narrower. I agree. But sometimes we have a scleroderma paper coming up in a few months or something. We needed to hunt down like really rare population, which we like almost couldn't see detect in the like with RNA without something like 10 or 12 fluorophores. It was like almost impossible. But I agree with the general notion of taking like broader gate. I think it's an important issue.
Thanks. Thomas, do you have any comment on that regarding how to work with sample types?
Yeah, maybe on the sorting, one approach we sometimes use is, for example, as an input for single-cell RNA-seq. For example, we sort and enrich for the rare population to get, let's say, I mean, anywhere between, let's say, 200, 300, 400 cells or maybe 1,000 cells, but then kind of fill up the rest with, let's say, total live cells. This way you still enrich for a target population, and you get information of the remaining cells kind of that are around either in that tissue or may it be peripheral blood. So this is one, I think, way to do it, to sort and enrich for a population, but then spike in also some just live cells.
Sure.
And generally, kind of, I agree with Jonathan that, you know, every tissue is different, and I mentioned that in my presentation. And most preps are geared towards immune cells, but even there can be quite a lot of differences in terms of what immune cells do you get out. Is it, you know, tissue resident T cells, or is it tissue resident myeloid cells you're interested in? But there are also like good online resources where one can look up how you can digest tissues. And this, for example, from Waddington, this is a very good website where they have actually species-specific digestion protocols.
Great, thanks, Thomas.
When I quickly jump in, Mandovi, I wanted to remind the audience that we have just 4 minutes left to go to our close. So if you have any questions, please send them in right now. I also wanted to remind you that there's a resource tab. If you click on the resource tab, you'll have access to some PDFs that have been uploaded there. So please check that out as well. All right.
We have a couple of questions here. So any input on library prep, Thomas?
I think when it comes to library prep, actually, let's say most of the experiment has already been done. So let's say if you do RNA sequencing, you have already reverse transcribed and amplified. So I think this last step, like for library prep, when you add the sequencing adapters, is almost like the easiest parts. And there are many commercial kits around that you can use. And I think probably just once you're done with library prep, at least what I'm doing is I do on each sample a quality control. So run a bioanalyzer profile or a tape station profile, because you really wanna sequence good quality samples.
Sure.
And I think this is important to check.
But I guess for any of these technologies that we talked about today, even if from the traces, from the bioanalyzer or the tape station traces, you can only say whether the library looks good or not. It rarely says, how good your data would be, because in my experience, a lot of time, if a sample has low viability, you cannot really tell from the library prep, because the libraries often look quite good. But then you don't know the answer to it until you get to the analysis part.
This is true for some samples, but take, for example, ATAC-seq, so an assay that looks for autochromatin regions. There it's actually, the bioanalyzer traces can tell you almost to 90% accuracy if sequencing will be good or bad, because it's a very distinct profile, and you can really see from the profile and judge if sample quality is good. I agree for other systems that this can be more tricky, but then, for example, one option would be, before going into library prep, to also check the amplified cDNA, how that one looks.
True, agree. Jonathan?
No, I just want to agree with Thomas regarding the QC. Sometimes we have in our protocol, we have two QC points. The most important one is actually we're doing one quality control before the first amplification of the library, and this is the most important one because it's like reflects exactly what we have there in that sense. And the second quality control is just before sequencing, and there we see how well it was amplified. So we're measuring a lot of things in these quality control tests. One thing is with which quality of initial material we started this process, and second thing, another thing, but the second thing is how well it was amplified. Anyway, yeah, super. I mean, it's super expensive to sequence, so you better have like good quality of material before you do.
All right. In terms of best practices for library prep, I would just like to add that, especially for single-cell RNA sequencing, you need to use a low-bind tube, and up until you get to an amplification step, you have to be careful not to lose any material along the way.
Mandalvi, can I ask you a question?
Yeah, sure.
I was just wondering, what are the common mistakes that you see people making? You work with so many different labs, so many different types of samples, and I was just curious. And I know you work only with single cell, but maybe some of them might apply to the bulk, sequencing as well. So, what do you see?
Especially, I can talk about single cell with more confidence than I can talk about the bulk experiments. So in my experience, people always rush to their experiment, and which often is, leads to disaster. So, it is important, since these are very high financial undertaking for any lab. It is important to do your due diligence in optimizing the protocol, and I know that people often try to, prep 8 samples at the same time. And from my personal experience, I can tell that even after working on so many different samples for past few years, I can tell that it is not trivial to prep 8 samples for single-cell RNA sequencing at a time. So I would rather personally focus on, say, 4 samples at a time and do it correctly. And this actually can save you a lot of money later, because what people do not realize sometimes is that, this bad quality sample can lead to a lot of read loss during sequencing. And that might cost somebody like huge amount of money and time during analysis.
And are there any resources that you refer people to usually before they come in to talk to you?
Resources there are lots of paper out there on sample prep. There is a good resource in protocols.io, which is a repository of many protocols, and there are a lot of protocols out there on many different sample types, which one can consult.
Oh, interesting. Protocols.io.
Yeah.
Now, of course, BioCompare also has a lot of articles and tech notes and things like that. They are also a useful source. But yeah, I think there's so much information out there that sometimes it's a little overwhelming, frankly. Just try to figure out what's really the most relevant. But did you have any other questions that you wanted to ask? Because we are kind of up on the hour, so if you have other questions, definitely we can continue.
No, I don't see any other question in the inbox.
Okay. Well, thank you so much, Mandovi, for moderating this webinar, fielding all the questions. Really, really appreciate your help. Thomas, thank you for joining us from Vienna. Good luck with your work. Just listening to you made me realize how complicated it is what you're doing with all the different organs and trying to do a sort of a systems analysis with different types of sequencing. Good luck is all I have to say.
Thanks for having me. It was great fun.
Oh, good. Thank you. Thanks. And Jonathan, same. Thank you for joining us from Israel. I know you're working towards your graduation. Working towards developing some new techniques which might change the way people look at things, not just looking at surface markers, but also working with intracellular markers, trying to identify cells based on their cell state, cell identity. I have so many questions for you actually based on the kind of work that you're doing, but this is very interesting. I might have a follow-up to this webinar, either in the form of an article or an infographic or something else, just to keep the interest going in this topic, because there's always so much happening here. So thank you again for joining us.
Thank you so much for having me. Thanks.
Yeah, and thank you to the attendees for being here. Again, wanted to remind you about the 3 resources that you can download and you can take a look at. And again, this webinar would not have been possible without the support of our sponsors, 10x Genomics, Roche, Beckman Coulter, Eppendorf, Levitas Bio, and Selecta. So I thank them for making this event possible for everybody around the globe. The event will be archived and will be available on BioCompare's website, which is biocompare.com, and a link to the archive will be emailed to all the attendees in a day or 2. So please look out for that email. And that brings us to the close of this webinar. Thank you all again for being a part of this event, and we look forward to seeing you at our next event.